somatosensory system, we've been focusing on touch reception. So my plan is we have like a slide or two more to finish off the touch story, and then we're going to be moving on to pain, which is maybe more fun. So we'll talk pain today, and maybe a little on temperature. There's only a slide or two on temp, but we'll hopefully get to touch on that as well. Friday, the plan is we're going to be talking about the Chapter 12 homework. And that'll be our focus for that class. Aside from that, so after break, I will get this quiz, and my plan is to get that quiz written. If I don't get it done before seminar, which is at 3, I will have it up tonight, but it will be appearing today. I've just been unable to get it done due to other things before now. But I'll get it up today, and then it will be due the Monday that we get back. I also decided for this quiz, I'm just going to do like multiple choice or matching for all the questions. So I haven't quite figured out exactly, but it won't be a fill in the blank for the Canvas quiz for Chapter 7. For the exam, certainly I think you should be ready and able to do fill-ins if that is how they are presented. Let's see. Oh, and then what we're doing after break also is we'll be doing, really there's only one more chapter in this unit. It's kind of a smaller unit. The somatosensory system chapter is the biggest, I think, and there's a lot of info in the somatic sensory system chapter. But what we'll be doing when we get back is a little bit of Chapter 11, which is the chapter right prior to this one, and that's going to be just a bit on vestibular system. So we're only doing the vestibular system bit, but I think that fits very nicely with the somatic sensory stuff, so it gets us sort of like this overall view of what our body is doing between the touch reception, the pain reception, and then the vestibular equilibrium system effectively. So we'll be focusing on that. And that's, that's a relatively small amount of material, but it's important, I think. And that'll basically finish off what we're doing for the next exam. So that's coming up. Any questions about anything? Y'all ready for break? I'm ready for break. Okay, so this is where I remember us being. We were talking somatotopy of the somatosensory cortex. So this idea that different parts of our body, the touch reception from different parts of our body, will go to slightly different spots along this same gyrus. So it all arrives at the postcentral gyrus, which is the location where we do have our primary somatosensory cortex. It's just going to different locations depending on where it originated. And it is a sort of distorted map of our bodies. So it's certainly kind of chronological is not the right word. I don't know what the right word would be. Somatotopy is what it is. In that we start at the foot and then we kind of move out to the arm and then we get the hand down here. So it kind of makes sense spatially, but it is distorted. And I did sort of ask you to think about why would it be that there would be so much more space dedicated to processing sensory input from the hand and sensory input from the face than sensory input from, like all basically the rest of our bodies, which is kind of a confusing thing on surface. There's a whole lot more space on my torso, for example, than there is on my face, and yet we have more brain area dedicated to processing info from our face than we do from the torso. The answer, I think, I could probably ask you guys, but I'm just going to spill it. The answer is that really what you're doing is that you're dedicating brain space in proportion to how many sensory receptors are coming out of these areas. So on our torso, so this whole stretch of our body down into our legs, we just really don't have that many sensory receptors. Each one has a fairly large receptive field, but there's a fairly small number of axons. I don't know why I'm touching my arm. That's exactly the wrong thing. There's a fairly small number of axons coming from swaths of our back, for example, that are conveying sensory information. We just don't have a whole lot of receptors out there. So you can tell that something's touching you, but you can't really localize it, beyond maybe to a patch that's about this big, because you just don't have that many axons that allow that level of distinction. So you don't have much resolution out there. So if there's not that many axons coming in from that big, large space, you don't need a whole lot of brain space to process it, right? I mean, you're just basically dedicating amount of brain space in proportion to how much sensory information is coming there. There's not that much sensory information coming from your back and your torso and your thighs and these kinds of locations. In contrast, like your fingers are just loaded with sensory receptors. We have incredible fine touch discrimination out on our fingers. That's why people that practice can read Braille, because they have that incredible level of precision, and that means in order to achieve that level of precision, we have to have many, many axons coming from each fingertip that we have. So the fingertips in particular are ridiculous, and then even, you know, our palm is very, very sensitive, and maybe not to the degree of fingertips are, but it's getting there. So our whole hands just have tons and tons of sensory receptors in them, and therefore tons and tons and tons of axons coming out of them, and therefore we need tons and tons of brain space to process that information. Similarly, our face has lots of sensory receptors as well, especially you'll notice around the mouth. And I think a big part of that is that we need to have that level of touch discrimination and touch information, I should say, coming from our lips and our tongue. Here's tongue down here, because that allows us to feel what our mouth is doing, and we need to feel what our mouth is doing in order to properly articulate speech, among other things. Certainly we want to be able to chew without biting our tongue and such as well, but I tend to think speech requires incredibly fine, and if you've studied speech, and especially speech pathology, if you've ever even done any reading on it, there's just not a whole lot of difference in terms of where exactly your lips are and exactly how tense they are to produce different kinds of sounds. And the same is true of your tongue. And they all have to work in concert in a very precise way, so you need to be able to feel what they're doing. And you know that you need to feel what they're doing, because if you ever go to the dentist and you have a numb mouth, all of a sudden you're like, not to mention you're slobbering all over yourself, but you can't talk. Like you're, you know, kind of just stumped. And that's because not because you've lost motor control, it's because you don't have any sensory input, so you don't know what you're doing. So I think that's why the face has so much information coming from it, is I think in large part due to our language needs. I guess that's my hypothesis. I don't have data to support that, but it sure makes sense. Okay, so that was kind of area mostly 3B. So that's all of our touch information coming into that somatosensory cortex, and it does show somatotopy. What this slide is showing is that the neighboring areas along that post-central gyrus that are associated with area 3B, like area 1, for example, which is depicted here, which is along the same gyrus. It's just like the cells that are right next to the area 3B. They are also exhibiting somatotopy. So it's kind of hard to, you have to like stare at this for a while, I think, to make sense of it. But what this is trying to do is trying to identify in area 3B the location of different body parts and where those body parts are mapping onto area 3B. And this is maybe a little bit more of an accurate depiction than the prior slide in terms of exactly what it would look like. So each of these little squares that you're seeing, or shapes that you're seeing, would be different structures. I think these are the toes. This clearly is the sole of the foot. Here's the leg coming in. But up here, you know, we've got thigh information coming in, trunk information, arm information. And then here's basically your arm and your hand coming in. And then these would be fingers. So each of these little spaces, this would be where we're mapping. So this is the same thing that was shown on the prior slide for area 3B. But right next to it, we have another set of cells, just like on the same gyrus, slightly posterior to them. This is area 1. Area 1, we said, was sort of in charge of receiving texture information. And area 1 also shows somatotopy. So the cells that are in this space right here are getting thigh information, just like these cells over here were getting thigh information. These cells are getting trunk information, just like these ones were. And if we go down to the fingers, what you can see is that, you know, so we have a whole set of cells that are also receiving touch information about the fingers over here in area 1. These guys are diagnosing texture information primarily in area 1, whereas these are more just sort of like generally receiving and then sending signals on from area 1. But they're still showing somatotopy. And so we see somatotopy in area 1 and area 2, and even a little bit in kind of like a rough sense in some of the downstream areas as well. So we continue to kind of isolate different bits of sensory information and confine it to different locations based on where it came from on our body. It's kind of a mirror image. So you can see, so this is area 3B, this is area 1, and so these are supposed to be the finger processing areas in area 3B and 3.1. This is kind of like just like a conceptualization of how it would look going left to right. So in area 3B, we have sort of the center of the palm is right along this margin between the two areas. and then we stretch over into the fingers such that the distal part of the fingers, the end of the fingers, is over here in area 3B. But over here in area 1, it's kind of reversed. It's like the mirror image. So the distal tips of the fingers would be over here. So that does mean like that the I don't know, sort of the start of the palm touch area is kind of right neighboring, are basically right next to each other in area 3B and area 1. It's kind of a fine point. But the main point is that we are still continuing to see this kind of somatotopy. Now this is an owl monkey, which is part of the reason that they're drawing the fingers to look kind of like monkey fingers, because they literally are. What they've done in this case is that they basically did experiments. This was a series of experiments. These date back into the 70s, where they would effectively just stick electrodes in different parts of the brain, and then they'd have of an owl monkey, and then they'd have the owl monkey's hand there, and they just sort of touch different spots along their hand or different parts and other parts of their body, and try to figure out what part of the brain was being stimulated as a result of that touch happening over on the different body parts. And this is the map that they created as a result of that kind of an experiment. Okay, so we have gone into the cerebral cortex. We're in area 3B. We've relayed up to areas 1 and 2, which is giving us some specialized cells that are specific to different kinds of textures, and so that therefore they may relay to other parts of the brain and help us to process texture information. And area 2 is giving us some size and shape information. And from there, we tend to relay back into more the posterior parietal cortex. So if our post-central gyrus, this green one here, which is where we have areas 3B, but also areas 1 and 2, and we've been kind of ignoring 3A, but it's in here too. All those areas are right along here. That's kind of like the most anterior part, the most forward part of the parietal cortex. All the rest of it we kind of collectively just call posterior parietal cortex. That's not to mean that there's not any compartmentalization here, but for our purposes that works fine. But posterior to this post-central gyrus, we have several different areas that are continue to be involved in processing touch information. So touch information kind of comes in to your primary somatosensory cortex in area 3B. It gets relayed to local areas like area 1 and 2, and then also gets relayed back into area 5, which is shown kind of in red, and area 7 back here, which is shown in purple. And these are doing a little bit more sophisticated processing. So we're continuing to have finer and finer discrimination and analysis of the kinds of information that are coming in. You'll see lots of really specialized cells in these areas that only respond to very specific kinds of input, like from one location with one kind of texture and one kind of shape. And so, you see a lot of specialization out in these areas. And this is probably where our sort of conscious perception of touch lies, where we can actually sort of perceive things. It's probably way back here in the posterior parietal cortex. Another thing that happens as we get out into this area is we get an intermingling of different kinds of sensory input. So, we're talking about touch information, and certainly we would also have pain and temperature information coming back here from the somatosensory cortex. But we also have other kinds of sensory modalities that we're getting information. So somebody asked about taste the other day. That potentially could have input getting back here. The visual system is kind of located back here in the occipital lobe, the very back of the brain. But information from the occipital lobe gets relayed up into the posterior parietal cortex, so we can kind of mingle our visual input and analysis with our touch input and analysis and sort of try to stitch those together in sort of our view of what's going on in the world around us. Similarly, like hearing and olfactions kind of coming from this area around this margin between the temporal lobe and the parietal lobe and the frontal lobe. So those signals can sort of go from here back into the posterior parietal cortex, where again you get this sort of multimodality processing that's occurring out there. Excuse me. Oh, and I forgot to, I meant to mention this also. So again, as we're going from the somatosensory cortex back into these areas, we get more and more sophisticated, more and more specialized cells. And so this was just this sort of direction stimulus is another cool thing. So just as another example of the degree of specialization, you have some cells that exist even in areas 1 and 2 that if you like take an object and you brush it from your fingertip down to your wrist, there'll be some cells that get really excited in those areas. If you do the exact same thing with the same stimulus, but you go from your wrist out to the end of your fingers, though, some of those same cells that were excited before won't be excited then because they're direction specific. So like you're getting all this specialization in terms of your processing. Last little bit that I think is kind of interesting, there's if you have lesions that occur within your parietal cortex, because you have all this sort of multimodal processing that happens up there. And again, this is where at least some of your sort of conscious perception of all of your sensory input probably lives and is manifest from. If you have damage up in here, that can result in some really interesting sort of just kind of puzzling sensory deficits or cognitive deficits that can occur in the people. So one of my favorite is something called neglect syndrome, or sometimes it's called contralateral neglect syndrome, where effectively a person just kind of stops paying attention to part of the world around them. So, and this sometimes it's called stereognosia too, where so like in this case, they asked a person to, here's the model that they presented them with. So they've got these different objects, and they said, all right, draw these different things. So they're looking at the object, and then they draw the things, and this is what this person sort of produces. So here's a clock, and you know, a lot of it's there, and it's not so much that it's not a perfect circle, but you know, it's kind of missing a bunch of stuff over here, right? Here's their depiction of the house, and it's just kind of missing the left side. Here's a picture of a flower, and it's missing the left side. And this was a consistent problem with this particular patient. They just seemed to not be aware of the left side of at least their visual input or visual information that's coming in. And what probably has happened there is that they had damage to the right side of the brain. And the right side of the brain is processing, generally, the sensory information coming from the left side. Now, I know this is visual input, but there is visual information that gets up into the parietal lobe as well. Let me finish the thought. You can also have situations where this is sort of manifest in terms of the body awareness too. So people that have contralateral neglect syndrome, for example, might only shave like half their face. And they kind of don't really just recognize the other half of their face as part of them. It's just like not there. So they'll just sort of leave it. And they just kind of neglect that half of their body. And the other half of the body, yeah, you know, they'll sort of shave down. But then there's just like nothing else to shave by their awareness. It's not that they just don't even really see it or think of it as their own, or these kinds of things. Barry, I'm sorry. I don't know. I don't know if it's the same. Yeah, I don't know all the vocab for the different things. Unilateral neglect? Similar kind of a thing? Mm-hmm. There could be, and I do not know them. But, you know, so yeah, so I'm gonna just say yeah. But the point of it again is that if you because you have kind of a lateralization of the processing of the sensory areas, if you have damage to one side, that's manifest then in sort of these cognitive deficits occurring that we're witnessing here. I don't know. I haven't really heard of it with smell. Yeah, I've not heard of that, like a lateral sort of issue with smell, but that doesn't mean it doesn't happen. I'm not positive, but I think that can be the case because, so certainly in terms, I haven't heard for pain, and that's why I'm hemming, but certainly for touch information, they may not perceive if you're touching that object. They just, that leg, they just don't perceive it. And I would imagine the same thing could happen in terms of pain perception. I asked you guys to read a chapter from this book for the homework. It's the chapter from this book. It's a great book. It's just full of like bizarro, weird cognitive deficit stories. So the book is called The Man Who Mistook His Wife for a Hat, which is a wonderful title. He literally was trying to like grab his wife and put her on his head at one point because he just couldn't like perceive differences in objects in space. He could hear her and talk to her and like he knew she was there, but he didn't like tactily and visually he couldn't perceive that. He just didn't like, he didn't understand it. Like, it's just like it didn't, he couldn't understand how to distinguish different kinds of objects. He could see them. Like, you could hand the guy a glove. This is not even the story I was going to tell you. But you could hand the guy a glove, and, sorry about that, he could describe, well, it's got a pouch with these other pockets, and he could even like propose ideas like you could put different coins in the different slots within the glove, but he couldn't recognize the thing as a glove. He just like lost the ability to recognize objects. He could recognize sound still. Yeah, he could see everything and feel everything, but he couldn't like make the processing step to recognize things. And so again, that's like posterior parietal cortex. The story relevant to your question, Danielle, though, was that there's another chapter of this guy that like wakes up in a hospital room and looks down and thinks that somebody put a severed leg in his bed with him, and he tries to throw it out of the bed, and he ends up throwing himself out of the bed because it's his leg. It's just he doesn't recognize it as his own anymore. He doesn't feel anything from it. It's like disconnected because he lost touch input, proprioception, like all that kind of information coming out of that leg. Sometimes I would think I should have you guys read that one instead, but I like the one I had you read too. Yeah, I think it definitely can. So stroke patients can't, like if the part of the brain up here gets damaged, I don't know about that specific example you said, but if a stroke patient has damage or deficits related to the stroke that are in this area, it can lead to some of these types of cognitive deficits for sure. Like in that particular case, I don't know if that's like they're aware that they're doing it, or if it's just, you know, poor muscle control, or I don't know, but yeah. I think I've heard of cases where it's recovered and cases where it hasn't. And I think it probably is specific to the person as far as what exactly has happened there and how bad the damage is. So I've heard of, I guess not this one, but I've heard of similar weird kinds of recognition deficits happening to people like post-concussion. And so while they've got, you know, post-concussion syndrome, sometimes people might have a hard time recognizing faces. It's just like they can't recognize people anymore. It just doesn't make sense. Like they can see people's faces, but they can't recognize. And I had a student who was describing this once. She had this for a couple days, and then it subsided, and she recovered, and it was okay. But I've heard of other people that have that problem, and they never recover from it. So exactly like, you know, probably has to do with how severely damaged the neurons are. Is there cell death going on? I'm waving hands, but that, you know, I think that's the answer. This stuff fun. I love this stuff. I should have been a psychologist. Anything else on this? That's kind of the story I wanted to tell on touch. What I want to do now is turn our attention to pain. Pain story is not quite as long as the touch story. In a lot of ways, it kind of mirrors what happens with touch, but the pathways and the anatomy is a little bit different, at least until we get to the cerebral cortex. So let's talk pain. How does pain work? I think pain is interesting because if you think about all the other kinds of sensory modalities, there's a way in which what you're perceiving in all the other cases, to me, is something a little bit more real than pain. So like with touch, you're perceiving pressure, mechanical disturbances placed on your skin. Like that's clearly a physical thing that exists in the environment. With sound, you're perceiving mechanical vibrations in the air. That's like a physical thing you can measure with the machine. Like that's, you know, that's a clear thing you're measuring. Vision, you're measuring you're measuring electromagnetic radiation, which is really cool that we can perceive that. But like, we can measure that with a machine. It's like there's a really clear physical thing that we're measuring there. Pain is not really that. Like pain is this perception that basically something has gone wrong and we are injured in some way. It's not directly measuring, I mean, sometimes we have some receptors that can key in on it, but it's not directly measuring like the injury itself necessarily. It's more measuring like how badly disrupted have our cells become. And the result of that is that we tend to have most of the receptors that are pain receptors respond to a bunch of different kinds of stimuli. We have some that are unimodal, but a lot of them are what we call polymodal receptors. So let me step back. So the general term for a pain receptor is a nociceptor. And so these are basically just receptors that are, that's the general term for a receptor that picks up pain information. The way they do so can vary dramatically depending on the nociceptor. So you will find some that are what are called unimodal. And so there maybe you might find one that's only responsive to just like severe mechanical stress. So if it detects that you've had like severe amounts of pressure or distortion placed on your skin, it might send a signal that you would then interpret in your brain as pain. Others are sensitive to chemical stimuli. So I think a nice way to think about this is that a lot of them are responding to chemical stimuli that only occur when cells nearby have been broken. There's a homework question on this, but a good example of this is, so some are sensitive to ADP. So ADP, this is the stuff that you use that your mitochondria turn back into ATP after they pump protons and have them come back across the ATP synthase. The only time you have a lot of ADP circulating around outside of cells is when cells nearby have been destroyed and ruptured, and so they leak a lot of ADP around. So we have pain receptor neurons that have receptors that will bind ADP if it's circulating around outside, and when you bind that ADP, that sends a signal up to your brain, hey, you know what, you've been hurt. There's some injury there. Some of them are temperature sensitive. Like I said, a lot of them are actually polymodal, so that would mean that the same sensory endings would have several different kinds of receptors. Some might be mechanoreceptors detecting pressure, and then on the same neuron, you might have some chemical receptors that might be keyed into different kinds of chemicals. You might have others still that are sensitive to temperature. There are some temperature receptors that double as pain receptors, so they kind of do both and send both kinds of information up. Once you get your receptor stimulated, it will send a signal up some manner of a fairly small diameter axon. So if you remember that diagram earlier in this chapter of different classes of axons, so we had the A alphas, which we mostly don't have anyway in skin, but we then had A betas, which is what all the touch receptors are. Most of the pain reception is going up either A delta or C fibers, and they then, so as a result of being fairly narrow diameter, and you might also remember that C fibers aren't even myelinated, so here's a picture of an A delta, and it's got some light myelination to it. Here's a C fiber, it's not even myelinated, but they're all very narrow in diameter. They don't send signals very quickly. We do tend to have, because we're using both of these kinds of fibers though, kind of two volleys of signal. I know we've said this before, but this graph is showing this. So the idea is that if you have a noxious stimulus introduced, so a noxious stimulus would be basically a painful stimulus, and then you sort of record the time course of when you receive different kinds of information, you would get touch information, which isn't even shown here, almost right away. Again, this is the example we always use as like a paper cut. So you would get that touch information almost instantly because those are coming up A beta axons super quick. You don't know if that's going to hurt or not, but you can kind of say, oh, maybe that's going to hurt. And then the first actual pain that you get would arrive via these A delta fibers, and it would happen pretty quick. You get a pretty, almost instantaneous arrival of that information. Not quite as fast as touch, because these are narrow diameter and they're not as heavily myelinated, so they don't have as high a membrane resistance, but they're still pretty quick. And then there'd be kind of a lull, and then you get, half a second later, you might start to get the second pain, as your book is labeling it here. And this is more like sort of the dull, aching, chronic, sometimes fairly intense pain that happens afterward. And that's primarily traveling up C fibers. So it's not a super fast signal to arrive, but it does explain sort of why we get sort of multiple volleys of this information coming into our system. Yeah. Ooh. So visceral pain, like stomach ache pain, I think is still following similar kinds of systems. In fact, I think I can get there pretty quick. So this area of your somatosensory cortex is getting intra-abdominal sensation, and pain isn't arriving at the exact same spots, but pretty close nearby. So that would be about where it would be, like visceral pain. Headaches, I don't know if I can speculate on exactly what's going on there. Like migraines are weird, and not all headaches are migraines, but still, I'm not sure it's the same system. I just don't know. Yeah, so like nerve pain, I think what you mean by nerve pain is where the pain is not really originating from an injury per se. It's more that the nerve that's conducting the axons from an area are inflamed or otherwise being stimulated. And so yeah, what's happening there is that you are getting sort of inadvertent firing and stimulation of these fibers like somewhere along their axons where the nerve is, and that's then leading to an interpretation in your body that there's some kind of a pain. We do a thing in anatomy physiology lab where we have you stick your elbow in a vat of ice water, and it's meant to be painful, like non-harming but painful. And what will usually happen is you'll start to feel pain up in your fingertips, even though your fingertips aren't actually stimulated there at all. They are outside of the water. They're perfectly warm, but your ulnar nerve conducts signals from your pinky and your ring finger, and that's going right through your elbow right there. That's your funny bone. And so that results in this kind of referred pain, which is kind of nerve pain. Okay, so we've got, we'll talk more about the receptors a little bit on the homework, which I think is a cool story. That's Friday. But we've got stimulation coming up axons now. And we understand there's kind of two different circuits, but they're all kind of going in the same direction. So we're going to follow these axons, A deltas or C fibers that are coming in, and try to follow their pathway in through the spinal cord and up into the brain. So we're mirroring what we did with the touch reception. So the story starts off mostly the same as touch reception. So we stimulated some axons. There's action potentials traveling up fibers. They're showing a C fiber. A deltas do the same thing though. So we go through the spinal nerve. We go through the dorsal root of the spinal nerve out in the rootlets. But then immediately we start to run into a difference. And the big immediate difference is we don't immediately have axon, at least axon branches or collaterals that get into the dorsal column. We're not going into the dorsal column at all. In fact, usually what happens is almost immediately we form a synapse between this primary afferent, the first order afferent, and the second order neuron down here in the spinal cord. And usually it's going to be occurring right here in this area that's called the substantia gelatinosa. So what can happen is, and there is a purple arrow here. So what will happen is the axons come in, and sometimes they'll kind of go up and down a level. But again, almost immediately, they'll form a synapse within this area called the substantia gelatinosa. So I'm right here. And again, you may go up and down a little bit in this zone of Lesueur, I think is how you pronounce it. But we're forming a synapse in the spinal cord. We haven't gone to the brain yet. And this is kind of the end of the line for the first order neuron. It does not go all the way up to the medulla. It is ending here in the spinal cord. That then gives rise to a second order neuron. The second order neuron, just like we saw in the touch pathway, it's the second order neuron that decussates and crosses from one side of the central nervous system to another. But the difference here is we're starting our second order neuron in the spinal cord. And so it then will cross over and get to the opposite side of the body. So if we came in on the left side of the body, we're going over to the right side up here. I should have said it the other way around, but that's the point. We're crossing from one side of the body in the spinal cord. And then the information will be carried up this tract. So instead of going up the dorsal column tracts up here, we're going up this area. This is called the spinothalamic tract. And so this is going to be an ascending band of white matter that travels and conveys pain information up to the brain. And we're already crossed over. We're already in our second order neuron before we even get out of the spinal cord here. Yeah? Yeah, pretty much all of them are. At least coming in from skin. Yeah. And that includes these guys. Okay. The synapse here, couple of details about what's going on here. So the synapse between the first order neuron and the second order neuron, primarily the main neurotransmitter it uses for that synapse is glutamate. It says this right here. So glutamate is an amino acid-based neurotransmitter. It's one of the main stimulatory neurotransmitters that is used in our central nervous system. We did talk about it back in chapter 5, I think. So it's glutamate is its main deal. But if you have a really intense pain stimulus coming in, you will, A, increase the amount of glutamate that you release for a more painful stimulus. But if it's really, really bad, you will also at the same time release substance P. Substance P we talked about as well. This is a peptide-based neurotransmitter. It's really like the main one that we're going to talk about. So this is multiple amino acids long, 10, 20 amino acids long. It will also be released from these same, at this same synapse and bind to a different receptors along this synapse. But that basically allows us to realize that we have just like really severe pain that's going on here. So that it says on the slide here, it promotes hyperalgesia. Hyperalgesia just means hypersensitivity to pain. So we're kind of just like amplifying everything whenever substance P is being presented. You probably heard of fibromyalgia, and I'm not going to claim that there's not some sort of that there's not a lot of debate about fibromyalgia. There are some physicians you talk to that they're pretty convinced that it's just like not even a real thing, that it's almost just people being overly dramatic or something. But there are studies that have shown some connections, and one of the studies that I've seen did indicate that there was roughly a 3-time concentration of substance P for a similar level of injury in people that had fibromyalgia. So what this means is that for a given pain stimulus that you're receiving, assuming that study is correct and that I haven't seen it validated subsequently, I haven't seen it not validated either, but assuming that's true, then what that would mean is that we're kind of just, for a given level of pain, if you have fibromyalgia, because substance P is being released more aggressively, you will perceive it as worse pain than it maybe actually is. But that doesn't change your perception. Like you're feeling that pain at a higher level than a standard person would. In general, people do release substance P, though it's not just fibromyalgia. You release this when you have moderate or intense pain, and so this is one of the things that we can do in order to perceive that. And of course, this is good, right? So like nobody wants to be in moderate to intense pain. That's not a happy thing. But the point of being in moderate intense pain is that it does tend to cause you to avoid using body parts that have been damaged. And so you tend to baby things, and therefore you allow them to heal and recover from pretty severe damage. I run. I'm not a good runner, but I run, and I always kind of joke, like, if you want to make small talk with me, you can ask me about whatever my current injury is, because I can always have something I'll be able to tell you about and complain about. But like what that does is that does require me to slow the hell down and not be stupid about my workouts, because I might have some elevated substance P going on, along with inflammation and other sorts of things that can exacerbate pain. You have a question? You know what? I said that, and this is the first week since January that I'm actually feeling pretty good. So February was a lost cause. It was a catastrophe, but that's all right. Well, yeah, that didn't help, but I don't know. Anyway, yeah, a lot of knee issues lately. That's okay though.
So, with like inflammation or swelling, does that also activate?
Yes. Yeah, so pain and swelling will activate pain receptors. It doesn't do it here. It would activate it down at the level of the tissue. But then that'll result in more action potentials being sent up here, which means probably that you're going to release more substance P along with your glutamate if you've got inflammation going. Because what the inflammation does is it'll release, among other things, you'll have bradykinin being released, which is a pro-inflammatory compound. And we have pain receptors that bind bradykinin and then communicate, hey, there's bradykinin here, I must be inflamed, I must be in pain, that kind of thing. Plus the swelling, just swelling alone can cause stimulation of mechanoreceptors, which are, some can be nociceptors, which can send pain input too. So yeah. We should all talk about our injuries. You can tell us about yours, and I'll tell you about my bursitis and whatever. All right. That's the other great thing. I teach anatomy physiology, so I'm like constantly diagnosing which tendon or whatever I'm hurting at a given moment. Doesn't mean I know what I'm doing, but learning my anatomy one injury at a time. Excuse me. Okay. So where we were. So just to pick up where we were. So we went in through the spinal nerve. That was here. We went through the dorsal root of the spinal nerve and through the rootlets. And then we formed a synapse. I don't know why it's showing this far down. It really should be showing the synapse way up here in that substantia gelatinosa. The right word? Yes, substantia gelatinosa layer. I was getting confused with the thing in this intervertebral disc for a second. So the synapse is occurring here. We're giving rise to a second order neuron down here in the spinal cord. It decussates immediately now. So we're still in the spinal cord and we've decussated. We have not gotten the medulla yet. And now we're going to be traveling up the contralateral side of the spinal cord from where the stimulus started up into the brain. So again, if you think back for a second, in the touch reception pathway, we went up the ipsilateral side, the same side of the spinal cord. So touch information came in from the right side and it went up the right side of the spinal cord. In this case, if we come in through the right side, we go up the left side of the spinal cord. So that is a substantial difference, and it can matter, as we'll see in a little bit. The second order neuron is going to keep on traveling all the way up, and it's going to go straight through the medulla, bless you, and finally end up in the thalamus. But like pretty much all other sensory information that's going to the cortex, we are going to form a synapse in the thalamus, and the synapse is going to be between the second order neuron that started down the spinal cord and cells, the third order neuron that begins in the thalamus. The thalamus then, in turn, and I should say this is coming into a couple different nuclei. One of them is that same ventral posterior nucleus that we saw before. But then there's another nucleus that gets involved too. It's called the intralaminar nucleus. These are just like little locations within the thalamus. But this is where the synapse would occur between the second order and third order neuron. The third order neuron would then carry us up to the primary somatosensory cortex, that S1 area 3B area that we were just talking about. It ends up in the same basic location, just in a slightly different layer and slightly different collections of cells in that area. So I keep harping on this, but I like this diagram. This is where I meant to focus on it. So this slide is trying to compare the pathways that we traveled in to go up through the spinal cord, up to the cerebral cortex. In the dorsal column medial lemniscal pathway, that's our touch pathway, and the spinal thalamic pathway, that's our pain reception pathway. Okay, so this was our first thing that we talked about last time. This is the thing we're talking about today. And so again, just to sort of trace you through, we come in this case, let's say we're coming in, so it's your side. So if we're coming in from the left side, if it's the touch pathway, we do form some synapses right away, but that first order neuron travels all the way up the ipsilateral side of the spinal cord to the medulla. There it innervates a second order neuron which decussates. Second order neuron goes to the thalamus. Thalamus goes up to the cerebral cortex, and then we get our signal in the primary somatosensory cortex. In this one, again, we're coming in from the left side, but we form a synapse pretty much immediately upon entry with the second order neuron. The second order neuron decussates, because that's what second order neurons do, but then travels up to the thalamus and then to the primary somatosensory cortex. So I kind of just walked through differences, but you can think for a second about what differences do you see between the pathways. I'm not actually going to ask you for your answers on that, but think for a second on it. And then consider this. I think this is kind of fun. So suppose that the left half of the spinal cord was damaged, but the right half of the spinal cord was okay. So there was a damage to the spinal cord, but it only happened on the left side, and assume it's just like total damage, total loss of the left half of the spinal cord. But the right half of the spinal cord is okay. This is something called Brown-Séquard syndrome. So if your left half of your spinal cord was damaged, what deficits in touch perception would you expect to see, and what deficits in pain reception would you expect to see as a result of that damage? Sort of puzzle it out for a sec. So basically the answer is going to be, are you losing touch perception below the site of the injury on the left side or the right side? And same with pain reception. Are you losing on the left side or the right side? So what do you think for touch perception? Which side should you lose touch perception on if the injury is on the left side of the spinal cord? Left side of your body? Agrees? Touch perception? You're saying right? So I got a left, I got a right. Well, I know. So like, okay, we're gonna have to vote. So who thinks damage should be on the left side? Touch perception. Nobody wants to take it. So 1, 2, 3, 4, 5, 6, 7, 8. Hands are popping up. I got 9 maybe. Who thinks the damage, the loss of perception should be on the right side? 1, 2. At least one person did. 3. All right, so we have like 13 or 14 people being willing to take a stand, and then everybody else is saying, no, I'm not going to. Okay, so let's say this is the left side of the spinal cord, right? So this is left, this is right. So if the damage is here, that's going to mean that that's going to interfere with this axon. This is an axon that came from the left side of the body, is trying to get up the spinal cord, and the damage is right here on the left side of the spinal cord. So that means that your perception, at least below the side of the injury, your perception of touch below on the left side of your body should be impaired, because touch travels up the ipsilateral side of your spinal cord. So it'll be wherever the damage is, you should have loss deficit on the same side of your body. That's touch perception. But what about pain perception? What do you think for pain perception? That's this side. Should be loss. So again, damage is on the left side of the body. Or I'm sorry, damage is on the left side of the spinal cord. So where's your pain loss? Left side or right side, do you think? A couple people say right. Getting some nods. Yeah, so damage is on the left side of your spinal cord. So that's over here. So that means that this information, this is from the left side of your body. If this is pain information from the left side of your body, it comes in, it decussates and travels up the right side of the spinal cord. So the left pain information, pain from the left side of your body, should be okay. It should travel right on up. But if we had the opposite drawn in, pain information from the right side of your body decussates, comes up, and then is going to get disrupted by this damage on the left side of your spinal cord. So if you have damage to the left side of your spinal cord, you should lose pain perception coming in from the right side of your body and touch perception coming in from the left side of your body below that site of injury. Which, like, nobody wants to have happen in there, but it's pretty cool. Like, you know, and this is something that, you know, this condition of Brown-Séquard syndrome is a case where somebody has that sort of a deficit. It'd be weird, right? Like, you can't perceive one side of your body, but you can feel pain from that side of the body. Like, bizarre. Yeah. That's a great question. Maybe. And I'm going to show you one place where there's a really overt interaction, though, between pain and touch pathways, and it would happen before you ran into the spinal cord damage. So it wouldn't surprise me if your pain is a little bit hypersensitive on the side where you lost your touch, but some of the interaction shouldn't be affected, should still happen. But yeah, that's a really good question. Brooke, and I'll go over there. Yeah. Uh-huh. So if the damage is on the right side of the spinal cord, is that what you're saying? I want to make sure I'm hearing what you're saying. So if I flip this diagram or this one? This one. So if I flip this, so, but the damage is still on the left side of the spinal cord, we're saying, right? So if that's true, so here is pain information coming in, it would decussate on a second order neuron and try to travel up this damaged side of the spinal cord. So yeah, if there's damage on the left side of the spinal cord, right side pain information should be lost, should be obstructed, yeah. Oh, so phantom limb, I think phantom limb is mostly, my understanding of phantom limb is it's mostly once you get up into the cortex. And we are going to talk a little about it, I think, on the homework. That's a pretty good stopping point. So what we need to do is we need to talk about modulation of pain. So that'll be Friday, the first thing. Little quick ditty then also on temperature. There is a question on your homework that talks about modulation of pain and endorphins and such, so you might have to read ahead a little bit. We'll talk about that at the start of class also on Friday, though. All right, I'll see you guys then.